Hey guys, welcome back to vaccine designing course. So today let's do the reverse vaccinology now. So first of all, make a folder named Immunoinformatics in your PC. Okay. So here it is. I have made a folder named Immunoinformatics. Okay. So here our aim is to do reverse vaccinology. Okay, so let's start with the steps. So the first step is primary protein structure prediction of antigenic protein. Okay, so we want the primary protein structure of antigenic protein, so we are gonna predict that. Okay, so as you can see in the slide, these are the steps that you'd be following. Okay, so primary protein structure prediction of antigenic protein is what we'll be doing first. So what does it mean by primary protein structure? It means the linear sequence of amino acids in a peptide or a protein. So to predict the primary protein sequence, the step that is done is retrieval of it. Correct? You need to retrieve its sequence. So here let's retrieve the linear sequence of amino acid. Okay, so the first step, as you can see in the slide, is to retrieve antigenic protein sequences. Okay, so the first step that you need to do is go to NCBI protein database. Okay, so again, as I have mentioned in the slide, to make you understand the step-by-step process better. Okay, if you want, you can take a screenshot of it. Anyway, we'll be providing you the slides and the notes. What I mean by notes is the short notes of it. Anyway, in the slides everything will be mentioned, and I'll also explain the steps one by one. If you have any queries, you can just ask to me. Okay, that's about it. So let's first go to NCBI protein database. So this is the link that you need to go, or you can either search in Google for NCBI protein. Okay, then you can click on the first result that it shows. But let's not complicate things. You just go to this link. Okay, so here either you can go to that particular link, or what I meant is you can type NCBI protein and the first result. So let's just take this. Okay, yeah, so this is the page that we'll be doing the prediction of antigenic protein sequence. Okay, and here whatever antigenic protein sequence that you need to figure out, you just have to search for it. Let's take, for example, search for MERS in the search box. Okay, MERS. Okay, what is it? It is the Middle East Respiratory Syndrome related coronavirus. Okay, so let's just search for MERS and then press the search button. Now let me just have a brief description of what MERS is. MERS, or Middle East Respiratory Syndrome, is a viral respiratory infection that is caused by Middle East Respiratory Syndrome related coronavirus, or MERS-CoV. Okay, now here you can see there are lots of results, right? Items 1 to 20, that is in the first page there are 20 results, but there are a total of this much. So this depends. Okay, this varies when it gets updated year by year or monthly. Okay, this is not the actual number. It again, what I meant is it gets updated. It'll always increase. Okay, so here you need to go for any result other than the first one. As you can see, first is FAD dependent monooxygenase in this case, right? So the first one, as you can see, it's of a different species. For me, or for this case, what we need is MERS, and the source what we need is human. Okay, so since it's streptomyces, we are negating it. Okay, so okay, you just go for any result. Anyway, I am gonna take one of the results and show you the page. So I am gonna take chain S spike glycoprotein. Okay, I'm just taking this as an example. There is no particular reason of why I'm specifying this, just that since I have done with this protein sequence, I'll be sure that I'll be able to take classes with this. Okay, you can take any one of it and try. Anyway, I'll be assigning you task for that also. Okay, so let's just open this 38th result. Chain S spike glycoprotein. Okay, so here as you can see, the source is beta coronavirus England 1. I think I made a mistake by saying human as the source. Let me just take the first result. I was telling about the first result, right? Let's just take the first result. Okay, 1 second. Yeah, so I'm taking FAD dependent monooxygenase. Yeah, if I take this, the source is streptomyces. Our source is again viruses. Okay, not human. I just said human. I'm sorry. Okay, for my interest of doing the study, my source I want is coronavirus. Okay, a virus. So that's why I negated the first result. Okay, let me just go to you can always take the second result, no issue. Yeah, so here in the second result, the source is Middle East respiratory syndrome related coronavirus. Okay, you can do further studies on that, but that you'll be doing it as a task. Okay, so let me just take the result of my interest. Okay, so it's chain S spike glycoprotein. Okay, so this is it. Now what we need is the protein sequence, right? So what you need to do is keep the cursor in FASTA. Here you can see identical proteins, that is protein related to this, then FASTA and graphics. Okay, so anyway, when you scroll down, you can read the information about it. It's locus, it's source. So this is our hero. Okay, organism is beta coronaviruses, and it says about the family of it, the authors, and rest everything. But what we need is FASTA. So let's click FASTA or right click it and open the link in a new tab. So then you will see the FASTA sequence. Okay, so it's a spike glycoprotein. So what next you need to do is copy this short sequence and paste it in Notepad. Okay, that's what you need to do is. So this you need to copy, then you open the Notepad and paste the sequence. Okay, so I have opened Notepad. My Notepad is ready. Now let's copy the whole thing. So this is what is called as the FASTA header, which says about its locus, then its chain ID, and then which type of protein it is. So let's copy the sequence as well as the FASTA header, open the Notepad and paste it. It's just Control-C and Control-V. Okay, now you need to rename this and then put it in your folder. Okay, which I've already mentioned to make a folder named Immunoinformatics, right? So now File, Save As, and save, either you can save as protein 1 or any other thing. So let's just save as protein 1 and in brackets spike glycoprotein. Okay, so in the desktop there is Immunoinformatics folder. So it's .text, it's in text format. So I am naming it as protein 1 in brackets, save as protein 1 and okay, 1 second, protein 1 spike glycoprotein and save it. When you see in the folder, you can see, okay, let's delete this. You can see that protein 1 spike glycoprotein which has been saved. Okay, so now the protein sequence is saved in the Notepad. Okay, so this is how you retrieve your antigenic protein sequence. Just have to go to NCBI database and take the FASTA format of your antigenic protein sequence of interest. Now, next task of yours is to similarly save the other results. Okay, you do one thing. Again, you do this exercise, search for MERS, and then when you search for it, you have first 20 results here, right? So we are not taking the first result, the reason which I've already mentioned it. So take the other 19 results. Similarly, save the other 19 results in different notepad files and then name it as protein 2, 3, 4 and so on. Okay, so that's what you need to do. So you'll be having different notepad files. That's our first step, okay, to retrieve antigenic protein sequences. So these were the steps that we did. Let's go to the second step, which is prediction of functional properties of antigenic protein. Now that we have protein sequence, next is we need to check its functional properties. If it's stable, does it have antigenic property, other properties and everything. Okay,